Welcome to the fourth webinar of the Cystatin C webinar series. My name is Shanyil Wickramasinghe, and I am the U.S. product specialist for Gentian Diagnostics. In this edition of the Gentian webinar series, I'll be going over two real-world scenarios that illustrate the clinical value of incorporating Cystatin C into medication dosing and monitoring protocols. In each of these cases, we will delve into the challenges faced by the organization, the solutions proposed to address the underlying issue, the outcomes of the implemented solution, as well as the value gleaned from the new dosing or monitoring approach. It should be noted that the data presented in this segment was collected from publications and independent research that will be referenced at the end of this presentation. The first scenario of this presentation is called Personalizing Chemotherapeutic Interventions through Co-reporting of Cystatin C and creatinine-based eGFRs. Before we delve into the challenge presented in this first scenario, it bears reminding that the kidneys are an important site of elimination for most antineoplastic drugs as well as their metabolites. In fact, most chemotherapeutic drugs, whether they be conventional cytotoxic agents or molecularly targeted therapies, are known to affect the nephrons and renal microvasculature with clinical manifestations ranging from electrolyte disturbances and proteinuria to acute kidney injury and progression of chronic kidney disease. For these reasons, accurate assessment of renal function is a critical component of both ensuring accurate dosage and preventing further renal decline in cancer patients. Unfortunately, most institutional dosing protocols and the very package inserts provided with chemotherapy drugs call for dose adjustments to be made according to the Cockcroft-Gault equation, which, if you recall, was developed in 1976 based on creatinine clearance data from a population of 249 male patients. Understandably, this equation has been described as being grossly inaccurate in certain populations and has largely been abandoned in favor of the more accurate MDRD and CKD-EPI equations. It shouldn't come as a surprise then that incorrect dosing of anti-cancer drugs that can occur as a consequence of using creatinine and the Cockcroft-Gault equation can lead to severe side effects, reduced treatment efficacy, reduced patient compliance, and in some cases, irreversible kidney damage. Faced with the challenge of developing a more accurate way to adjust cancer treatment doses, the hospital in our first scenario proposed implementing a system that involved the co-reporting of eGFRs based on both Cystatin C and creatinine for each patient receiving chemotherapy. In the event that a patient's Cystatin C-based eGFR differed from his or her creatinine-based GFR estimate by more than 15 milliliters per minute per standard body surface area, an automated alert will be generated, prompting an integrated care team to conduct a workup of the patient and create a personalized therapeutic intervention based on that patient's unique needs. It's worth noting that in this case, the investigators calculated creatinine-based eGFR using the modification of diet and renal disease, or MDRD equation, and calculated the Cystatin C-based eGFR using the chronic kidney disease epidemiology collaboration, or CKD-EPI equation. Of the 606 patients receiving cancer treatment at the hospital, the investigators found that 193 of them would be classified as being in chronic kidney disease stage 3A and 3B. And of these 193 patients, almost 20% were misclassified as to which stage of kidney disease they were in and were therefore at high risk of receiving the incorrect dose of a potentially nephrotoxic drug. Diving further into the results of their investigation, the care team noted that incorporating Cystatin C testing into kidney function assessment allowed them to prevent CKD misclassification and improve dosing accuracy of antineoplastic agents in 25% of patients with severely impaired kidney function. Theoretically, this meant that side effects would be avoided in 1 out of every 14 patients undergoing cancer treatment at the hospital. Avoiding side effects like nausea, fatigue, and anemia also has the secondary effect of improving patient quality of life, which then increases the likelihood that the patient will remain compliant with the prescribed therapeutic regimen. This in turn is said to have markedly improved physician confidence and satisfaction levels within the oncology department, as they would no longer be burdened with the need to manage treatment side effects. From a purely financial perspective, developing personalized dosing regimens for the 5 medications listed here, nivolumab, oxaliplatin, carboplatin, cisplatin, and trastuzumab, generated over 104,000 euros in annual savings through dose reductions. What's noteworthy here is that the added cost of incorporating Cystatin C into the evaluation of the 606 patients involved in this project was around 1,800 euros. The second scenario presented is called Cystatin C-guided dosing of vancomycin, a novel approach to achieving trough concentrations among ICU patients. As the title of this scenario aptly describes, this next segment of the presentation describes the clinical benefits of using Cystatin C to guide dosing adjustments of vancomycin. Briefly, vancomycin is an antibiotic with a narrow therapeutic index, which is commonly administered intravenously to treat serious gram-positive infections. Concentrations of vancomycin above the therapeutic threshold are associated with nephrotoxicity and ototoxicity, while concentrations below the threshold can lead to treatment failure and risk of developing bacterial resistance. The challenge facing clinicians is that only an estimated 20% of patients actually attain a vancomycin concentration within their individualized goal range under the current standard of care. Like the dosing of anti-cancer drugs in the previous scenario, the standard approach to adjusting the vancomycin dosage involves an assessment of kidney function based on the Cockcroft-Gault equation. Because more than 90% of this drug is eliminated by the kidneys, an accurate assessment of renal function plays a critical role in ensuring treatment efficacy and safety. The significance of this issue from a public health perspective is also clear when we consider the fact that vancomycin use has increased more than 100-fold over the past 40 years due to the rise in incidence of community-acquired MRSA infections. The challenge facing the medical team administering vancomycin is illustrated by this graphical representation of the dosing process. We see that the therapeutic window for vancomycin lies between 10 and 20 milligrams per liter in this figure. When the patient receives the initial infusion or the loading dose, the concentration of the drug rises rapidly and begins to fall as the drug is metabolized. Once the concentration falls to the initial trough level, the patient is then given a maintenance dose that's calculated according to the rate at which the drug is cleared, which in this case depends on the patient's kidney function. Depending on the condition being treated, additional maintenance doses of vancomycin will be administered following the same process for as long as it is necessary. And while steady-state trough concentrations of the drug are ideally reached by the time the third dose is administered, ensuring that the trough concentration remains within this narrow therapeutic range depends largely on how accurately the clearance rate of the drug is calculated. And so, to optimize the vancomycin dosing process, the care team proposed implementing a new dosing algorithm, one which incorporated Cystatin C measurement into the calculation of vancomycin clearance rate. Specifically, the new algorithm would use the CKD-EPI creatinine Cystatin C equation to estimate glomerular filtration rate instead of the Cockcroft-Gault formula. And to compare the performance of the two algorithms, investigators conducted a prospective study that evaluated the steady-state goal trough achievement between ICU patients whose doses were calculated according to the current standard of care, using the Cockcroft-Gault equation, and patients whose vancomycin doses were calculated according to the new Cystatin C-based dosing algorithm. What investigators found was that while only 28% of patients dosed according to the current standard of care achieved a trough concentration within their goal range, 50% of patients dosed according to the new Cystatin C-based algorithm were within their goal range. What's more is that patients receiving the antibiotic according to the new dosing algorithm achieved a steady-state trough concentration 25% faster than patients following the standard approach. This has broad implications for the level of care received by patients because earlier identification of inadequate vancomycin concentrations would allow the medical care team to either increase the dose or determine whether an alternative gram-positive antimicrobial would be needed. Going back to the underlying public health concern of antibiotic overuse and the need for antibiotic stewardship, it's also important to note that under the Cystatin C-based dosing approach, patients required 7% less vancomycin before trough assessment. Now, although investigators of the original study weren't able to characterize a relationship between Cystatin C-guided dosing and specific clinical outcomes, I'd like to point out that other studies have found that patients experiencing nephrotoxicity from vancomycin experience, on average, a 3-day increase in the median hospital length of stay. Additionally, studies have also shown that vancomycin-resistant enterococcus infections are also associated with a 13.8-day increase in the average length of stay. And finally, in terms of the direct monetary benefit, we estimate that the increase in Cystatin C test volumes generated from the implementation of this novel dosing approach would reduce Cystatin C reagent costs by as much as 25% per test. In summary, we've learned that using Cystatin C-based eGFR to estimate renal elimination of medications leads to improved dosing accuracy, better patient outcomes, shorter length of stay, and significant cost savings for the hospital. Before we end this edition of the webinar series, I'd like to remind you that Gentian's platform agnostic Cystatin C assay has been both CE marked and FDA 510(k) cleared as of 2008. It's also been standardized against the human serum certified reference material for Cystatin C, which was developed by the International Federation for Clinical Chemistry and Laboratory Medicine. The random access workflow capabilities available on most automated platforms also means that this test can be performed with lead times as low as 10 minutes. The references to the information presented in the course of this webinar are provided below. For more information about Cystatin C and the Gentian Cystatin C immunoassay, please contact us at marketing@gentian.com. Thank you.